Hi everyone, we are from Long Island University and we'll be presenting our research on antibiotic production in Central Park. Just to introduce ourselves, my name is Saji and I'll be presenting with my peers Minna, Abdelaziz, Aman, Kaur, and Engy, Dous. For a little bit of background on why we chose this topic, antibiotic resistance is a major global issue and it's really significant because just in the United States alone, it's responsible for about 35,000 deaths. And pharmaceutical industries and drug developers don't really have much of an incentive to go after finding novel antibiotics because of high costs and a lot of other variables. So us as students and researchers, we think it's very important to go out of our way to study antibiotic resistance and maybe even look for novel antibiotics. We were able to go to Central Park and study wooded, meadow, and sandy trail areas to look for producers. We originally thought that the wooded areas would have more producers and more colony forming units, but we will talk about our results and our abstract later on.
Hi, my name is Aman and I will be speaking about the methods. So earlier this year in January, we all went to Central Park and collected 8 soil samples from 8 different locations throughout Central Park. The 8 locations all belong to 3 different substrate types: wooded, meadow, and sandy trail. The collected soil samples were serially diluted and plated on 3 media types: 10% TSA, BHI, LB, all of them including cyclohexamide to inhibit any unwanted fungal growth, and they were incubated at 30 degrees Celsius for 48 hours. After incubation, the colonies were counted and colony-forming units were calculated. After having calculated the colony-forming units, unique colonies were identified and further plated on master plates to make them more visibly comparable. Master plates were then incubated for an additional 24 hours at 30 degrees Celsius. And from the colonies that were incubated on the master plates, 247 colonies were screened for antibiotic production against escape pathogens to further determine the presence of antibiotic production through presence of inhibition zones.
So we were supposed to carry out our research and actually sequence the antibiotic producers that we had, but unfortunately, because of COVID, our research was halted. But we did yield some really great results that would help with future research. So just to draw your attention to the 2 figures that are on the screen, these 2 figures compare the colony forming units across the different location types and across the different media types. In the first figure, you can see that although there's a wide range for example, the sandy trail, all 3 of the means for the 3 different location types are approximately the same, with a p-value of 0.361, which allows us to conclude that they approximately had the same amount of colony-forming units across the location types. It was very similar with the different media types, where the means were approximately the same, p-value of 0.327, that allowed us to conclude that across the 3 different media types, they had approximately the same amount of colony-forming units. If we move on, you can see in these 2 figures over here, these 2 are comparing the percentage of producers now across the different locations and across the different media types. So in the first figure, you can see that now there is a significantly higher percentage of producers in the meadow than comparing the other 2 locations. And similarly with the media types, there was a significantly higher percentage of producers in the 10% TSA than the other 2 media types.
So our conclusion, we had to first reject our hypothesis because our original hypothesis was that the wooded area would yield a greater amount of antibiotic producers, and we assumed that the greater amount of bacteria found in the wooded area would yield this great amount of producers. However, we have to reject this hypothesis since we found out that the meadow areas had a greater percentage of producers in our case. Our objective in this study was to compare antibiotic production across different substrate and media types. And our results showed that the meadow areas had the highest percentage of antibiotic producers, but also the media type affected the antibiotic production. Our overall conclusion was that there was no difference in the number of colony-forming units in different substrates. However, there was significance in the amount of producers found in each area. So future studies should definitely screen for novel antibiotics through genetic testing. Also, they should consider looking for differences in antibiotic production over different seasons, weather conditions, and over longer periods of time. So this concludes our presentation, and we just want to say like a huge thank you to our our mentor for this project, Dr. Christine Zolnik, for making it possible and for guiding us. And also, we are extending our gratitude to Long Island University, the Biology Department, and the Honors College. Thank you.